All right. Hello, everyone. It's 7 o'clock. Thanks for attending the May lecture as part of the Arizona Arc and His lecture series. My name is Blaine Burgess. I'm the communications officer for the Arc and His board, and it's my pleasure this evening to introduce Jakob Sedig. Jakob is a coworker of mine at Chronicle Heritage and is also a co-editor for the journal Kiva. And tonight, Jacob's going to be presenting on ancient DNA in northern Mexico. I'll just remind everyone that throughout the talk, you're welcome to enter any questions in the chat function at the bottom of your Zoom screen, and Jakob will be addressing and answering those questions at the end of his presentation. Otherwise, that's all I have to say. Jakob, please take it away.
Right. Let me share my screen and start the presentation. All right, we should be good to go. I can't see anyone, but it's great to be here. I'm coming from the East Coast, so it's 10 o'clock my time. I made sure I had a cup of coffee before I started, so I wouldn't be falling asleep during the talk. But it's really my pleasure to talk to you tonight about this work that I've done in ancient DNA in North and West Mexico during my time as a postdoc in the Reich Lab at Harvard University. Work that's still ongoing and I'm still working to complete when I'm not doing my day job at Chronicle Heritage with Blaine and then also co-editing Kiva. So the format of the talk, I'm going to give an introduction, a little bit of background about me for folks who don't know me. Then I want to cover ancient DNA methods. I find that it's when I give these talks, it's helpful because ancient DNA is a newer technology that we've started to apply to archaeology. And there's concepts and terms that I think it's helpful to introduce so that we are on the same playing field when I'm going over these various topics. Then we'll talk a little bit about some previous studies that have been conducted in West and North Mexico and in general in ancient DNA. And then we'll talk specifically about the PIPANOM project and some work that I published last year with some co-authors about a child from Paquimé who had very closely related parents. And if there's time, there should be, I'll also give some preliminary results on big picture demographic trends that we're seeing from this large data set that we have from North and West Mexico. So 10 years ago exactly, I was doing Mimbres archaeology and barely knew what ancient DNA was. It was not on my radar. My dissertation research was at Woodrow Ruin in the Upper Gila of Southwest New Mexico, and I was happy as could be excavating, writing reports, analyzing data from the work that I did at Woodrow, and had no idea that I'd take a pretty major detour and spend close to a decade in an ancient DNA lab. But you never know where your career path is going to lead. And so I finished my dissertation, moved out to the East Coast with my wife. My wife is from Beverly, Massachusetts originally, and just happened to get connected with David Reich, who's the director of the Reich Lab of Medical and Population Genetics at Harvard University. And for a while, for a couple years, I served as the lab's consulting archaeologist. So that really involved establishing relationships with museums and other scholars and other communities and being sort of a interpreter of archaeology for the geneticists in the lab. I was the first the archaeologist in the lab. The geneticists there were doing really great, interesting genetic work, but when it would come to things like radiocarbon dating, they needed help interpreting that. And that was my job. And then David got a large grant that allowed him to bring me on as a postdoc. And I was a researcher postdoctoral fellow along with the lab's ethics and outreach officer from 2018 to 2023. And so I did a lot of work in the Americas, trying to understand the ethical landscape and best approaches for doing ancient DNA projects in the Americas. We built up some really nice collaborations with researchers in Mexico and Central America. And so the work that I'm going to be talking about is these projects that we've initiated and as I mentioned, are still ongoing. Um, so yeah, I transitioned from the field to the ancient DNA lab, and along the way have published on a variety of topics. Some of the papers that I'm most proud of include establishing ethical guidelines for ancient DNA research in globally. These basic principles that all ancient DNA researchers should adhere to. We did a study on African American descendants from an ironworks in Maryland a couple years ago. I've published papers on the Maya region. I'm not a Maya archaeologist, but I've had the fortune to learn all about Maya archaeology in this role, in my role in the ancient DNA lab. And then a really exciting paper that I'm going to talk about in a little bit, where we collaborated with Chumash community in California and some indigenous geneticists to study the movement of Mexican-like ancestry into California prior to the arrival of maize agriculture. And then, as I mentioned, most recently, a little less than a year ago, this paper where we found a child in Paquimé who had very high levels of consanguinity, which is the fancy term for inbreeding. His parents were very closely related. So to go over some basics, some ancient DNA basics. So when I'm throwing around terms like SNPs, you'll know what I'm talking about. First things first, ancient DNA, when I use the term ancient DNA, what I'm referring to is the DNA of an organism that died anywhere from not even hundreds of years ago, tens of years ago, up to hundreds of thousands and maybe even millions of years ago. There's no true age cutoff for when DNA starts to be considered ancient. Basically, it's when an organism dies, that organism's DNA starts to degrade, and at that point, it's when we consider it ancient DNA. So when we say ancient DNA, we're really talking about a set of techniques that are used to reconstruct an organism's DNA after it's died and its DNA starts degrading. With that being said, sort of colloquially, the way we use these terms, you'll see sometimes people refer to more recent DNA as historic DNA. So that Maryland project that I mentioned, which is from the Revolutionary era through the Civil War era, we would call that historic DNA, or a lot of people would call that historic DNA, versus ancient DNA, which is generally in the Americas reserved for pre-contact. But it varies in different parts of the world because of the different histories that different parts of the globe have. Studying ancient DNA is a very complicated process for a variety of reasons. Jurassic Park, the original Jurassic Park, the one that came out, I guess, more than 30 years ago at this point, there's a little snippet in the movie where there's a character called Mr. DNA, and he explains how they extract DNA from an embryo or an amber of a mosquito that has been preserved in the amber, and they basically suck out blood from the mosquito and make it seem really simple. It's much more complicated than that, though. With that being said, the concept is basically not too far off. One of the things they talk about in Jurassic Park is that the ancient DNA is full of holes, right? And in the movie, science fiction, they fill in those holes with the DNA from modern frogs. But the part that they have right is that ancient DNA is very degraded. DNA, the moment an organism dies, it stops copying itself, it stops replicating, and so it starts to fray along the edges. And depending on the preservation context, like anything in archaeology, you can get better preservation in some areas, in some regions than others. So The best ancient DNA that we found and the oldest DNA that's been recovered comes from really cold places, Arctic tundra. You can get DNA from hundreds of thousands of years ago. Heat, it doesn't really, as far as I'm aware, there's not too much of a difference whether it's hot and dry or hot and humid. Um, Either one is bad for DNA, and I'll talk about that a little bit more when I talk about Paquimé. But the more degraded the DNA is, the less information we're able to gather from it, and the more we have to do to reconstruct it. Typically the fragments that we're getting are very short. So if you know your basics of biology, you think of DNA, it's the double helix. It's a long chain over 4 billion base pairs long, the individual adenines, thymines, cytosines, and guanines, ATCG, all in a row. But that chain starts to break down, and so we'll get chunks, maybe 30 or 50 in a row out of those 4 billion plus. So we're really talking about small fragments. And on top of that, we have contamination. That's our other big issue, is figuring out whether or not a particular sample that we're working with has been contaminated from modern humans or other organisms, as the DNA has been in whatever context it's been in for anywhere from decades up to hundreds of thousands of years. So doing ancient DNA work, it is like something out of science fiction. When I started in the lab and was learning about this, was seeing pictures and going into the lab, I really couldn't believe the technical sophistication that it involved. People who have heard me give talks about ancient DNA have heard me make this statement before, but during COVID, working in an ancient DNA wet lab was one of the safest places that you could work, because we use all sorts of PPE and filtration systems in our wet labs to make sure that modern DNA doesn't contaminate the ancient individual that we're working with. So these wet labs, along with the PPE, the face masks that you can see here, and the bodysuits, the air only run one direction. It's constantly being filtered out. The room, when no one's in it, we use the UV light to kill modern contaminating DNA. So there's a variety of things that we do to make sure that the ancient sample isn't contaminated. We only need a very small amount of bone powder or whatever material. We're able now to generate DNA from all sorts of substances, like soil, even coprolites, chewed quids. Regardless of what the material is, we only need a very small amount if the DNA is preserved well. So I'll walk through those steps that we do, the DNA, ancient DNA analysis pipeline. So we start with whatever material we're working with, more often than not bone, but it doesn't have to be bone. We take that substrate, whatever we're using, into our clean room, and here you can see an example of the UV light on, which is killing any modern contaminating DNA that might be in the room that might contaminate the modern DNA or the ancient DNA that we're working with. If it's bone, we turn the bone, we take a very small sample from the bone, 37 milligrams, which is smaller than the tip of a pencil. We turn that into a powder. The powder is then, we take a number of steps to turn that powder into what's called an extract. It's basically liquefied. There's a number of steps that removes all minerals, any other material that is in the bone, leaving us only with the DNA. We turn that extract into a library, which is basically making copies of the DNA that are floating around in this soup that we've created. Then we take that library and put it through what's called a sequencer. The sequencer reads all the individual A, T, C's, and G's that are floating around, and then aligns them against a reference genome. And then we get the raw data, which is what I start to look at when I'm doing analysis. Now, I've picked up some skills along the way, but I wouldn't consider myself a geneticist. I'm an archaeologist. I was trained in how to dig stuff up out of the ground, not how to do these really sophisticated statistical techniques for analyzing ancient DNA data. With that being said, having been in the lab for so long, I've learned a few tricks of the trade, and I can do, I may be selling myself short. I can do the basics pretty well at this point. But the point is, once we have this raw data, once we start with whatever material we're starting with, and it goes through all the steps, we get some raw data that we put into our database that we can co-analyze with previously published samples or any other data that we've generated in the lab. So a few more things, a few more terms that I want to go over again, because I'm going to be using them. So as we're generating this ancient DNA, the majority of it is not coming from the ancient individual that we study. The majority of it is what we call non-target DNA. So in this little pie chart here, the target endogenous DNA, and 5% that's great if we get 5% target endogenous DNA. That's the ancient DNA that we're interested in from a particular individual. The rest of the DNA that is generated that's in with that material is non-target endogenous DNA. So that means DNA from other parts of the genome that's not as informative as the particular portions that we're looking at. Environmental DNA. So if you have a bone or a coprolite that's sitting in the ground for hundreds or thousands of years, it's coming into contact with the environment and picking up whatever might be in DNA from whatever organisms are in that environment, bacteria, for example. There's modern contaminants. So if we're working with ancient individuals and those individuals, those ancestors have been on museum shelves for decades or hundreds of years, they're very likely contaminated with DNA from modern individuals, people who excavated the ancient individuals, museum curators who have handled the bones, all of that, prior to the modern techniques of wearing a mask and wearing gloves, would have contaminated the ancient individual that we're interested in studying. And then taxonomically unidentifiable DNA. So I mentioned, we typically aim for chunks of the genome that are 30 to 50 or more of those individual base pairs longer, the A, T's, C's, and G's. But we get lots of small fragments that are only 2 or 3 bases, or 10. And that's not enough for us to determine, which organism that comes from based on those few base pairs alone. So SNP, I'll be using this word a lot. A SNP is a single nucleotide polymorphism. So the human genome, as I've mentioned, is over 4 billion of these A's, T's, C's, and G's. But the majority of that is what's called junk DNA. It's not informing us about anything in particular. So I'm sure you've all heard, all humans that are alive today and that have ever been alive are 99% identical genetically. That's true. We share a lot of our genomes, and the majority of that 99% isn't really telling us anything. A single nucleotide polymorphism is a position in the genome that is informative. It's a variant. It's where, individuals can vary from one another. So for the longest time in the lab, we targeted 1.24 million of those SNPs. We've actually expanded that recently where it's up to about 2 million SNPs. These variable positions on the genome that we study. And so when we're generating data from an ancient individual, very rarely do we get those full 2 million SNPs. If we have 100,000 SNPs, that's great, and that's enough for us to work with. But we basically, when we're doing our analysis, we'll have, 10 individuals, 100 individuals, or 1,000 individuals, and then anywhere up to 2 million positions in the genome that we're interested in, that we can compare to any across all the individuals in a data set that we're working with. So SNPs are really key. The more SNPs that we have, the better for the analysis that we're doing. It gives us more statistical power and more resolution. One other thing I wanted to mention is the difference between mitochondrial DNA and autosomal DNA. So the type of ancient DNA research that I've been involved with is nuclear DNA or autosomal DNA, and that really is maybe about 15 years that people have been doing that. Notably, one of the most notable studies is the when the Neanderthal genome was generated, and then Denisovans. But prior to that, people were doing ancient DNA research too, but that was almost solely mitochondrial DNA. So the mitochondria is an organelle in the cell that is outside of the nucleus of the cell, where the autosomal DNA is. And mitochondria has its own DNA. Now, the mitochondrial DNA is only 12,000 base pairs long. It's many fold times smaller than the nuclear DNA. But there's much more of it than nuclear DNA. That's why people have been able to do mitochondrial DNA for so much longer, is there's a lot of it, and it's not nearly as long and complex as nuclear DNA. The thing about mitochondrial DNA is that it's only passed down from mothers to their offspring. So my mitochondrial DNA comes from my mom, and her mom. But it ends with me. My daughter's mitochondrial DNA comes from my wife, from her mom. So any offspring that I have, they're not getting mitochondrial DNA from me. They're getting it from their mom. You may have heard, I don't know, this is when I was in undergrad, I think. So it's kind of scary to say, about 20 years ago, I guess. But, there's famous study about mitochondrial Eve, because this is only passed down from mother to offspring. You can trace this back to, what they call mitochondrial Eve, some of the earliest human ancestors. But again, we're talking about 12,000 SNPs compared to, well, really 2 million out of 4 billion for nuclear DNA. So we're able to do many more things with autosomal DNA than we are with mitochondrial DNA. Okay, so I hope that was a good enough crash course for you to at least understand my talk and some of the things that I'll go over in the talk. I spent years learning this stuff. It was like getting a second PhD, having to learn all the genetics and the biology. But I think with that, you should hopefully be able to follow along with the studies I'm talking about, and at least have a basic understanding of the terms when I use them. Okay, so a few key previous studies that I think will help frame some of the work that I've been doing. I wanted to mention this. This was a previous study of mitochondrial DNA from the Mimbres region, which is where my background is, and this is from 2011. Now I'll say, that sort of the obvious thing about this study is that this was done at a time when NAGPRA regulations were different than what they are now. In order to do this study today, it would require intensive consultation and engagement with Indigenous communities. But because of the way NAGPRA regulations were written 15 years ago, the engagement and the consultation that occurred was not what we would do today. With that being said, what this study found by examining mitochondrial DNA with some individuals from the Mimbres region was that, well, there's a couple interesting things. So we divide mitochondrial DNA into what we call haplogroups, which is basically a way of grouping all the different variants within those 12,000 base pairs in the mitochondrial DNA I was talking about. And we know from various studies of modern mitochondrial DNA that this haplogroup B is the most common in the US Southwest, and there's lower frequencies of haplogroups A, C, and D. Um, Haplogroup A in particular is more common in Mesoamerica. And when I say Mesoamerica, I basically mean south of Chihuahua and Sonora, especially as we get into Morelia and central Mexico. We see sort of a increase in haplogroup A and a decrease in haplogroup B. So unsurprisingly, these Mimbres individuals, haplogroup B was the most common. There were a few haplogroup A individuals, but again, that's expected. That's not, when we see haplogroup A, we're not saying that someone from Mesoamerica was in the Mimbres region, because we do have low frequencies of that haplogroup in the US Southwest. The potentially interesting part from this study was that there's an individual from Nan Ranch who had haplogroup B. Again, that's the southwestern haplogroup, but there's mutations in that haplogroup that's really common in modern day Tarahumara. And as we know, Tarahumara are from, sort of the south of the present day border. So this paper, was using this to say that, people, the people who lived in the Mimbres region may have connections with northern Mexico, Sonora, which is interesting when we're thinking about demography and what happened at the end of the Mimbres period, when there's been all sorts of discussions and debates about movement of people south from the Mimbres into places, especially like Paquimé, that I'll talk a little bit more about. But again, this is an early study. This is only looking at mitochondrial DNA, even though there's maybe some evidence of Mexico and Mesoamerican connections. Our resolution here is not what we can have with autosomal DNA. So I previewed this study a little bit when I was doing my introduction. This is a study that was published about 1.5 years ago that had a really interesting finding. This was published before the NAGPRA updates that came out in January 2024. But even prior to those updates, the amount of engagement and collaboration that we did with this paper was really extensive. So we worked with the Chumash community in Southern California. We had one of the authors, multiple authors on the paper were Indigenous geneticists, Indigenous archaeologists, including from the groups that were part of this study. So there was a lot of engagement, really meaningful engagement that occurred that let us do this study in a way that was respectful to the communities and help them answer questions that they had about their history. The results. This study, we studied dozens of ancient individuals from the Southern Channel Islands and the Northern Channel Islands, along with a few individuals from the PIPANOM data set. This is some of the first PIPANOM data that was studied. I'll talk more about PIPANOM, but PIPANOM is the big North and West Mexico project that I've been involved with for the last several years. One of the basic things that we do in any study like this is we combine our newly generated data with previously generated data, and we do some sort of basic demography to understand how the different populations relate to one another. What this is showing is basically there's nothing unexpected here for the most part. The groups that are in closest geographic proximity are more closely related genetically. The interesting part of this study, though, is we were able to study some ancient individuals that date back to 7,000 years ago. Those were the oldest individuals that we had, up until right before contact. And what this graph is showing is the increase in what we call Mexican-like ancestry in California through time. So the Mexican-like ancestry, we equate to Uto-Aztecan ancestry. And this is coming from some of the ancient individuals from the PIPANOM data set that we included in the study. So what you see here are the oldest California individuals have none of this Mexican-like ancestry, none of this, I'm a little hesitant to call it Uto-Aztecan language, but we're basically using it as a proxy for Uto-Aztecan ancestry. It's at 0 at 7,000 years ago. But by 5,000 years ago, we're already starting to see some of that Mexican-like ancestry in California, including up to nearly 40% in some individuals. If you're familiar with the discussions and debates that have happened or occurred about the spread of Uto-Aztecan language, one of the leading hypotheses is that Uto-Aztecan language spread from Mexico north into the Southwest and into California with the spread of maize agriculture. As far as we know, that dates to about 4,000 years ago. But we're seeing that Mexican-like ancestry in California about 1,000 years prior to the spread of maize. So this is sort of providing a it's going against what the traditional Uto-Aztecan hypothesis might expect. We see Mexican-like ancestry in California prior to the spread of maize agriculture. So that's really interesting. And then you can see, as we progress through time in general, that it continues to increase. We have more and more Mexican-like ancestry in Southern California as we move forward through time. So this really was a key finding in this paper was, that this ancestry may be related to Uto-Aztecan speakers is moving into the Southwest in California prior to the spread of maize agriculture. So it's showing us how genetics can help inform some of these long-standing questions and debates that we've had in archaeology. Okay, so now I'm going to spend a chunk of time talking about this case study from Paquimé that was not something that we were focusing on in particular for this project when we were planning the project. So PIPANOM, a little bit of background. PIPANOM, it's an acronym from Spanish. It stands for the Proyecto de Investigación de Poblaciones Antiguas en el Norte y Occidente de México, which basically translates to a project investigating ancient populations in ancient North and West Mexico. This is a project that is really spearheaded by our Mexican collaborators, in particular Jose Luis Punzo, who's been he's become a friend and has been a great collaborator on this project. We've done all sorts of outreach with local Mexican communities as part of this project, including a DNA day for school kids where they can learn about DNA. But this project is years in the making. We first started working on it in maybe the end of 2016, and it took about a year to hammer out an agreement between INA and Harvard. And then we started collecting the samples from across Mexico for this. COVID slowed us down. Various, other things slowed us down. But we're finally to the point where the paper's getting close to being done, and I can present preliminary analysis. And as I mentioned, a few of the ancient individuals from the PIPANOM project have been published as part of that California paper. So we wanted, when we were coming up with this project, one of the key things we wanted to do was to make a basic genetic map of ancient Mexico. We have all of these cultures, from northern Chihuahua down through central Mexico into Michoacan, that have interesting histories. And we, as archaeologists, we can see differences between the material culture, but we don't really know how biologically how the people related to one another. So we wanted to create this base map, basically. And in order to do that, we have to get, genetic samples that have both geographic and time depth. So we were able to collect samples from, 309 ancient individuals with working data from across Mexico, from the various cultures that we have pictured here on this map. Something I haven't talked about yet, and I don't want to spend too much time on it, is which skeletal elements provide the best genetic data. Long story short, the petrous bone produces better genetic ancient DNA data than any other skeletal element. I won't get into the reasons for that now, but that's the discovery that the petrous generates this really rich, robust ancient DNA data has allowed us to work in places like Mexico. Because prior to this, the data that was being generated from teeth or postcranial elements was not of good enough quality to allow us to do really sophisticated statistics to understand population demography. The Petrus has really allowed us to do the PIPANOM study. Our mean SNP count for these 309 individuals is about 460,000. That's great. If we get 100,000 SNPs, we can do genetic studies with fewer than 100,000 SNPs, but 100,000 is, if we have 100,000, that's good. We can do everything we need to do with 100,000 SNPs. So 460,000 is great. And then, as I mentioned, we take these new data and combine them with previously studied, previously published individuals, and so our working data set is over 500 ancient individuals for PIPANOM. I'm going to come back to this. You'll see this a few different times, but this is showing you that base map that we've been able to generate with the PIPANOM data. So what you see here are the various sites and regions coded by color and shape. So, moving north to south, we have I won't get into how we decided to group things geographically, but I'll tell you, we spent a lot of time thinking about this. So things are either considered, we either group them by broad geographic region, or if there's reason, specific site. So, we call sites from the north, we'll just label them north, and then either 1, 2, 3, 4, 5, based on their chronology, 1 being earlier, 5 being later in time. So you can see that here in the key, north 2, 3, 4. Paquimé, we're calling its own thing because we have specific research questions we're interested in about for Paquimé. CDLMC, that's Cueva de los Muertos Chicos. I'll talk more about this. These data have been published in the California paper. There's some really interesting things, though, that are also happening big picture geographically. Again, I won't spend too much time on this now, other than to say, when we are generating new data for the PIPANOM project, we feed it into this base map that we have now, and we do just sort of a quick initial assessment to see, if there's any outliers, if there's anyone whose genetics look different than what we might suspect for a particular time and place. If you squint, you can see there's a few, but in general, Paquimé, there's nothing that really stands out. This is just this is from the published paper from last year, focusing only on published data. This doesn't include the unpublished PIPANOM data. And what you see here is a lot of pretty colors, but what it's telling you is genetically, this one individual from Paquimé, burial 23-8, fits with what we would expect genetically for the genetic landscape of Mexico, pre-contact Mexico. What stands out is something that's called runs of homozygosity. Runs of homozygosity are parts of the genome that repeat themselves. And the reason they repeat themselves is because an individual has parents who are closely related. So if my parents were first cousins, they're going to have a similar DNA. And that would mean that the DNA that I inherit from them, there's going to be parts of it that repeat more so than someone who had parents that are not closely related. We can use that in a few different ways. One really interesting way that I'm not going to talk about now is we can use that to study the population size of an individual. Reason being, if someone's coming from a mating pool that's small and closed, and people are closely related or more likely to be closely related, they're going to have more of those repeating parts of the genome. Another way we can study it is to find out how closely related an individual's parents might be. And if an individual that we're studying has long sections of the genome that repeat itself, long runs of homozygosity, that's telling us that that person's parents were closely related. This individual from Paquimé, burial 23-8, when we look at the expected values for various types of relatives, it had parents who were more closely related than first cousins. This is the value that we would expect for first cousins. parents were not as closely related as siblings, but more closely related than first cousins. So those types of relationships would be half siblings, having parents that are half siblings, parents that are aunt and nephew or uncle and niece, grandparent and grandchild, as sort of maybe unsettling as that might be to think about. But those are the types of genetic relationships that the data from this individual is telling us, that how closely this individual's parents were related. The value that we found just through our standard screening process, this runs of homozygosity is now something that we do as part of our standard process. It jumped out as soon as we generated that data for this individual. And in fact, when we saw this, we compiled all previously published individuals from the Americas and found that those values, there's only one individual that has values that high, and it's this one individual from Paquimé. Most individuals from the Americas, and this is, from Canada down to Tierra del Fuego, have values that are between 0 and 125. The value for this individual from Paquimé was 282 or 283. Very closely related parents. That's not the only interesting thing. So I saw this data as a geneticist, but the individual, when I first looked at that individual, only had the lab's ID, the arbitrary lab ID that we assigned, I22220. So I saw this high value and immediately started doing some digging. And it's not just that this individual has high runs of homozygosity. He was also buried in a room from the House of the Well, which is a very ritually significant room at Paquimé. Not only was he buried in the House of the Well, he was buried beneath a post, wrapped around the post, the base of a post, on top of this stone disc. Underneath the stone disc were some turquoise pendants. There's no other burials that have been found at Paquimé like this, and as far as I'm aware, in the broader Northwest Southwest. This burial type is one of one. And in the Paquimé report that Charles de Peso wrote, he says this type 8, he gave it its own unique type. Type 8 burial represented the sacrifice of a young child, which was wrapped around the base of a large support post beneath the floor of room 21C8. The body position of burial 23-8 indicated that this child was placed as some sort of supplicatory offering at the time the post was seated during the Paquimé phase remodeling. What's not mentioned here is that the reason he calls it a supplicatory offering is because there's damage to the skull of this individual. Clearly there was a blow to the back of the head that killed this child. So this seems to be a child sacrifice. This isn't new. This is work de Peso did 50 years ago. But it's not a coincidence that the genetic data really stands out for this individual too. At least I don't think so. So, as the peels of the onion are coming off, we start doing, more and more research to learn as much about this individual as we can. I'm referring to the individual, or I'll refer to the individual as he, acknowledging that gender and sex don't always correlate. When I say he, I'm referring to genetic sex or chromosomal sex. Studying the sex of this individual, he has X and Y chromosomes, which again doesn't necessarily mean his gender while he was alive was male, but biologically that's the what we found was that he had X and Y chromosomes. So I refer to him just sort of as he, with all those acknowledgements that I've mentioned. But so we learned as much as we could about him. We did strontium isotope analysis and found that there's been a number of individuals from Paquimé that have had their strontium isotopes studied. And so we compared the value that we generated for this individual from those previously published individuals and found that he undoubtedly lived his short life at Paquimé. I should say too, the osteological research on this individual showed him to be 6 to 8 years old when he died. So he's a child. He spent his short life at Paquimé eating food, animals, plants that were from the Paquimé area. We did radiocarbon dating on this individual, and that part agreed with De Peso's assessment. De Peso notoriously, some of his dating has been questioned, but he felt that this individual was from sort of the apex of the Medio period, the height of the occupation of Paquimé, and that's what our radiocarbon dating found too. I'm already running short on time, so I'm going to move through some of this material pretty quickly. But as all of this is coming together, we started doing research on these runs of homozygosity values that have been published globally for ancient DNA. And at the time that this paper was studied, there was really only one other individual from Newgrange, which is a Neolithic shaft tomb, really cool site in Ireland. And there was an individual whose parents clearly were siblings, had a long ROH of 687. Excuse me. Again, this individual from Paquimé, burial 23-8, has one of the highest values that's been published in all of the ancient DNA literature. His parents were very closely related. It turns out that that's rare. We found that in the ancient DNA literature, but we've also found that in studies that have been done of ethnographic studies of different cultures across the world. And in looking at modern DNA data sets. It's very rare to find people that had parents who were more closely related than first cousins. And that's because most cultures have inbreeding or incest taboos. There's various reasons for that. There's been a lot of published work on sort of genetic abnormalities that can occur when two closely related individuals have a child. But there's also something to be said for taboos and who is allowed to break taboos. And I did a pretty thorough search of the ethnographic record once it was pretty clear that this individual had closely related parents. And what I found was that there's very few exceptions where inbreeding is condoned, and those exceptions almost always come from elites in hierarchically structured societies. Well-known examples being in ancient Rome or ancient Egypt, but in other societies that have some form of social organization, hierarchical organization, it's usually the elites that are allowed to break this incest taboo or this inbreeding taboo. And it's because in general, that's what elites do. They can do things that everyone else is not allowed to do. And so when you consider some of that evidence with Paquimé, maybe this is telling us something about the social structure there. I always have to throw this in. Game of Thrones, at least the original series, is now, it's old, maybe 10 years, more than 10 years ago when it first started airing, but it's another well-known example of incest or inbreeding among elites. It's not out in the open in Game of Thrones, but it is the elites who are inbreeding, at least in Game of Thrones. So what we argue is that this was a child of an elite lineage. His parents were closely related, and they had some special role at Paquimé. And he may have been sacrificed as a way to consecrate this special building, the House of the Well. That's the story that I've been telling, since this paper came out last August. There's an exciting addendum that has nothing to do with this study, but this is a new study that came out less than a month ago. And it fits very nicely with everything that I've been saying about this child from Paquimé and elite sacrifice and sacrifice of children in Mesoamerica. If you haven't read this paper yet, I highly recommend it. You may have seen press releases from it. It was published in Antiquity, the same journal where we published our inbred child from Paquimé paper. But at Tikal, well-known Maya site, they found an altar which clearly seems to be linked to Teotihuacan. And there's been all sorts of debates, as I'm sure most people know, about connections between Teotihuacan and Tikal. And there's just been more and more evidence of the past few years. And I'm not going to say much about the genetics, but it's an active question that people are working on in genetics too, in ancient DNA. But anyways, this altar that they found at Tikal clearly has motifs from Teotihuacan, and also there's some sacrificed children that were buried around the altar. One of the press releases says that this altar was used for human sacrifices, especially of children. And in that literature research that I did for the Paquimé paper about sacrifices in Mesoamerica, one of the things that I found was that sacrificing of children is something very powerful that people did, and it's disturbing to think about, especially as someone who has a 5-year-old daughter. Right? It's not a pleasant thing to think about, but it did occur in ancient Mesoamerica, and I think that's what we're seeing at Paquimé too. So again, it's I won't say it's nice seeing evidence of sacrifice of children, but this paper coming out after our paper in a totally different area, but providing evidence of a behavior that I think we're seeing at Paquimé too. It aligns with the work that we've done. Okay, I was telling Blaine before we started that I try to limit my talks to 50 minutes because I know I talk a lot and it's a lot to take in. And it's almost 11 o'clock for me, so I don't know how much longer I can keep going. I do want to do some brief, maybe spend 5 minutes going over preliminary results from the broader PIPANOM project and some of the interesting findings that we have before I open up the questions. So again, this is our culture area that we're looking at, trying to create this base map of what the genetic structure of ancient North and West Mexico looked like, and answer some of these long-standing questions about these interesting archaeological cultures. For the most part, the data that we've generated in this base map, what we see is that genetics correlate with geography, meaning as we move from north to south, or I should say south to north, based on this admixture plot down here. These are previously published Maya individuals. So they have sort of a combination of this different components of ancestry, some orange, some yellow, and some green. And then as we move, into Mesoamerica, we can see this orange predominate at Tzintzuntzan, a site Jose Luis Punzo works at. It's a really interesting site. We have many individuals from Tzintzuntzan, as you can see. And then as we move north, along the Sinaloa coast into Durango, we see an increase of this light green ancestry. This is the Mexican-like ancestry I was talking about with the California paper. And we can see in this PCA plot that individuals cluster, for the most part, pretty nicely based on geography. So orange here is the orange correlating with North Mexico. Green is Tzintzuntzan. Purple is the published California individuals. Gray is Maya. This is what we would expect to see, these broad geographic regions clustering with one another. We've made one of these nice trees that also show, again, nothing too out of the ordinary. If we're looking at Paquimé, the 50 plus individuals that we've studied from Paquimé are, very closely related to other individuals from North Mexico that are contemporaneous or predate Paquimé. I'm going to point out here Cueva de los Muertos Chicos and Trincheras sites individuals. Cueva de los Muertos Chicos is this site in Durango. Trincheras individuals are here. So opposite ends of what we call Arido, Mexico. So the arid part of Mexico. What's interesting is that Cueva de los Muertos Chicos is the southernmost site that we have in Arido, Mexico. But their ancestry, Cueva de los Muertos Chicos, is almost as the strongest signal that we have of that Mexican-like ancestry in our data set, even though they're the furthest south. Vice versa, if you look at Trincheras individuals, they're the northernmost group that we have, just about the northernmost group that we have. But their ancestry, they actually have a pretty significant orange component here, which is more of the Mesoamerican-like ancestry, the Tzintzuntzan ancestry. So there's something interesting going on here. It's not a one-to-one correlation. It's not this, a gradual transition. There's some interesting things going on here that I'm working on now, and we'll hopefully have a better sense of when we publish the PIPANOM paper. Um, there's a lot going on here. Basically, what this is showing us is an F3 statistic. We compare two groups to each other. So the darker blue shows any two particular groups are more closely related to each other. So again, individuals from North 3, so North 3 is our sites here in Chihuahua, are more closely related to Cueva de los Muertos Chicos, this site, because they're dark blue, they have a higher value than other sites from northern Mexico. Or Paquimé, yeah, it's about the same as Paquimé, but definitely more closely related than Trinchera sites, which is geographically closer than Cueva de los Muertos Chicos. So again, something interesting going on. What else do I want to say? I think I'm going to end it there. I could get into some more details, but those I think are the most interesting things. This clearly is still a work in progress that we're getting closer to publishing. It's important to acknowledge all of the people that have been involved in the study, most importantly, the ancient people that we study and their modern descendants. As I mentioned, for the PIPANOM project, we've done a lot of really meaningful work with local Mexican communities on this project. There's a ton of PIPANOM collaborators, archaeologists from across Mexico who have worked with us, sent us skeletal remains to study, ancient individuals to study. The Consejo de Arqueológica, INA, who approved this study and has been very supportive. The co-authors on the Paquimé paper, the inbred child that I mentioned, we've received funding, the Reich Lab in particular, from various organizations, Howard Hughes Medical Institute, John Templeton Foundation, which also funded my postdoc, and the National Geographic Society. Lots of thanks to the Reich Lab technicians, the people who work with the data and process it and get it to a place where someone like me can analyze it. And then Arizona Arch and Historical Society for giving me the time to talk tonight, and for electing me as one of the current editors to Kiva. I can't end a talk without making a plug. We need articles. So if there's anything that you're working on, let me know, and we can find a way to publish your article. And we are really emphasizing the types of articles that we haven't seen traditionally in Kiva. So, if you're in the CRM field, if you're doing work in historic period, those are the types of things, or regions for Nanamogion, that might not typically be covered in Kiva. I think it'd be great if we could increase those articles. So again, reach out to me. We are in need of articles, and if you're working on something and get it to us soon, in all likelihood, you can get it pretty quick turnaround for getting it published. So thanks, and I'm happy to open it up to a Q&A at this point. So I think the way we said this would work is that questions will start showing up, and I'll just read through them and answer them as they come in, if there are any.
Yeah, thank you for your great presentation, Jacob. Yeah, I just want to encourage folks to please drop questions in the Q&A section. I have one just to lead things off and spruce things up, but I couldn't help but pull out, David Reich's book while you were doing the whole DNA intro, right? And I just, kind of breezing through a lot of the studies that he gave examples of that where DNA augmented kind of archaeological information. I just wanted your take, Jacob, on the field now and maybe in the future. Like, what has been kind of the major contribution of genetic research and information to the field of archaeology and in the regions that you're studying, in the populations and the sites, et cetera, et cetera?
Yeah, great question. And yeah, David would admit, he says that book's outdated now. Genetics moves at a speed that I'm not used to as an archaeologist. Papers that are 2 or 3 years old in genetics are sometimes viewed as outdated. So that book, David is, I think would say that he's due for a second edition. it was interesting when I came into this. The things that archaeologists and geneticists were debating and getting into heated arguments about has sort of changed through time. But one of the big ones that I wasn't involved with but observed was the spread of Indo-European languages and Yamnaya culture through Europe from the European, from the steppe, excuse me, from the Eurasian steppe, through Western Europe. That was something that I saw a lot of debate about, and that was one of the things where archaeologists were very upset with what the geneticists were saying because it countered, years and years and years of archaeological research. That was an example of genetics and archaeology not correlating. Not correlating in a way that I think people expected. I think there's been multiple papers since then. This was 2017, around 2017, 2018 or so. And I think that things have settled a little bit since then. Maybe another example that I give that I've given is I went to the European Archaeological Association meetings in 2023 in Belfast, and it was really almost jaw-dropping to see the type of work that they're doing in Europe with ancient DNA now. So anytime there's an archaeology project, it seems, what will happen if there's burials involved? That burial will get full ancient DNA work, full strontium isotope work, full carbon nitrogen isotope work, full radiocarbon dating. And they're basically looking at these, micro scales of movements of people through time, looking at the movement of biological females or women. We can see in Copper Age Europe, they're moving much more frequently than males were. And so there's been all sorts of articles published about what that means and sort of maybe bride exchange or just matrilineal matrilocal systems that may have been happening there that have become obvious because of genetics, but also I guess because of the strontium data. But it's adding granularity to the archaeological record and previous hypotheses and shedding new light on things that we just weren't able to see before. There's lots of examples. I can keep going and there's been sometimes like in the Yamnaya example I was talking about where the genetics and the archaeology didn't correlate, but there's been plenty of other times where genetic data supports archaeological hypotheses. I don't. Either way is interesting to me. It's adding a richness to the stories that we tell from the archaeological record, and it seems like still every other week or every month or so there's a new paper that comes out that's having major impacts on our understanding of the past. I see a question came in. I also saw Paul Minnis's hand raised. Paul, I don't know if you still have your question. Paul's already, he's seen this talk before or a version of this talk. But so I don't, Paul, if you're still there, do you still have your question or should I move on?
I would just echo to folks that unfortunately I can't turn on the talking ability for the group, but you're more than welcome to put your questions in the chat or in the Q&A box at the bottom of the Zoom call. But yeah, I see have some have already popped in, Jacob, so I'll let you address them.
Yeah. So I had in the Q&A, there's from Anonymous, it says, when I did undergrad work decades ago, we learned about 3 major migrations across the Bering Strait. Has there been enough genetic work to expand on or refine that idea? The answer is yes. There's some really exciting work that's happening there. Some of the major people that have been working on that, there's Jennifer Raff. She's a professor at University of Kansas. S.K. Willerslev at Copenhagen are some people that have been involved with this work. So, I could spend a semester talking about this. I'll try to be as brief as possible. when I was in grad school, which is decades ago at this point, it was still pretty much the Clovis first model, meaning we didn't really have genetic data. But the most accepted model was people came over across the Bering Land Bridge at the end of the last ice age as the ice sheet started to melt. And you could move from, sort of Beringia down through, Alaska across the New World. Genetics correlate with that. There's not been definitive proof yet genetically of people being in the Americas prior to that model. Now, with that being said, there are some huge caveats. What we do know genetically, what we found genetically is there was a Beringian standstill. So as people were coming over from Northeast Asia, Siberia, when Beringia was a tundra before it was underwater, people spent enough time there that they started to diverge genetically. There's genetic drift from them and mainland, Northeast Asia groups. And then after the Beringian standstill, they moved, the ice sheets melted, they moved into the Americas. Those are sort of the foundation, those people were the sort of the foundational groups of modern day Native American people. There's been plenty of archaeology, though, I think that is more definitively showing that people were here in the Americas prior to that. But the thing is, genetically, we haven't found that yet. And there might be a couple of reasons for that. One is if there were groups that were genetically distinct, they may have been so small that we just there's no skeletal remains, for us to study to see that genetically. It's possible that if there were groups here, they were the Clovis model replaced those early groups. We don't see them genetically. I don't know. It's still sort of an open question about well, let me put it this way. Personally, I think there's enough evidence now that we can say that there were people in the Americas prior to the melting of the ice sheets. There's not genetic data, but there's plenty of explanations for why there might not be genetic data. So then you have that initial pulse, the peopling, the Clovis model, and then at a later period in time, there's a second migration of Athabascan people that comes in, in northern North America, Canada, and then moves down, through what today is the U.S. Genetically, that's pretty clear too. That's based on modern genetic work because there hasn't been really any studies of ancient Athabascan individuals for all the reasons that people might expect. Okay, so from Bill Doolittle, I see something in the chat. I am taken with the Mesoamerican contribution to DNA in the Trincheras region in contrast to the Casas Grandes DNA farther east. Will you be looking at DNA evidence from the area between Trincheras and Casas Grandes? The short answer is no, I wish. The longer answer is not yet. So as I mentioned, the PIPANOM project was years in the making, and we had planned to get data from many more places than we were able to prior to saying we need to stop and analyze the data we have and publish something. So down the road, we very may well have data from that area in between. I didn't present on any because we don't have any yet, but it clearly is an open question. What I'll say too, and I'm sorry if this wasn't clear, it's still unresolved as to why those Trincheras individuals have different ancestry than the Paquimé individuals. I don't necessarily think it's evidence of Mesoamerica coming up into Trincheras. It may be that those analyses still are ongoing, and we need to look into it more. But it's also possible that that component was more ancestral, and it's just been there for a long time, and wasn't fully replaced by Uto-Aztecan ancestry that we're seeing, in Paquimé and other parts of Mexico. So again, it's clear there's a difference. Why that difference exists, there's a few different hypotheses that we still are testing and trying to figure out. Lane, I don't see anything else.
Yeah, while I have one more question.
Sure.
Sorry.
Yeah. No worries.
But
Yeah.
this gives also other people any additional time they might want to use. Um, and if you have questions later on, you can always, okay, two people are raising their hands.
Okay.
So I see folks are raising their hands. If you could please drop stuff in the chat box or the Q&A if you want to be anonymous. But anyways, while you type those out, I just wanted to know if you had any thoughts on let's go to the House of the Well, on the actual context of that burial. Any kind of thoughts on it? Yeah, when you're talking about it, I think, okay, was this veneration? Is it veneration to be under the post of a house or whatever? Or did DePeso offer any context as to whether those were deposited at the same time, or was the individual laid after the fact? Because I think the top-down image was that the child was wrapped around the post, but the profile made it seem like I believe the individual was under it. So I was a little confused if you could add anything.
Sure. So when I've given this talk before, and when the paper was being reviewed, probably the most common question I have is, how do you know, one of the arguments we're making is that this child was, the offspring of two elite individuals who are closely related and sacrificed, I think, as part of a building consecration, like some ritual ceremony related to that. And so the follow-up question is, how do you know that this wasn't some sort of illicit relationship? And actually this child they were trying to hide the burial. That's possible. That I think that we'll never really know the nature of the relationship between the two people who had this child. But when you look at the sum of the evidence, like I've been talking about, it becomes, to me at least, it's all the pieces fit together. So that the burial in particular, that was reading DePeso and what DePeso says about the context and the date. It's pretty clear that individual was placed with the construction of that building. The burial was not placed at a later period in time. It's not like the building was built and then they came back, redug to the base of the pillar and wrapped the child around it. It was all part of this. While the building was being constructed, this happened. But then again, as you start to bring in these other lines of evidence, it's usually, not always, but when we find evidence of inbreeding, it's elites that are allowed, that are condoned, that are allowed to do to have children that are the result of two closely related parents. In Mesoamerica, we know sacrifice. We have to. I think I going back to my grad classes and the little that I know about sacrifice in Mesoamerica. The way that we might think of sacrifice or conceptualize sacrifice is not the way that the people who performed the sacrifice did. What it meant to them isn't what it means to us. And so the most powerful thing my understanding at least is the most powerful sacrifice that you can make in ancient Mesoamerica is your blood. And so if you're sacrificing a child of an elite family, that's going to be a very powerful sacrifice. And again, we know this building, there's all sorts of features about the building that I didn't talk about. The well itself, but it clearly was a ritually important room. And so if you have this elite lineage who's trying to demonstrate that they're very powerful, the most powerful sacrifice that they can make is, a child from this lineage, this powerful lineage. I know it kind of seems like a just so story, but when you look at the all of the data together, it's not just the genetics that we're looking at, it's the corpus of the data. I think that's how the pieces fit. Okay, a couple more came in. Okay, please remind to the UA contribution to Paquimé individuals. So this is referring to Uto-Aztecan contribution to Paquimé individuals. So again, it's not, when I say Uto-Aztecan, there really needs to be a detailed study that looks at how the language correlates with genetics, which isn't always one-to-one. Uto-Aztecan to me is kind of a stand-in for what I was calling like Mexican-like ancestry or North Mexico ancestry. And at Paquimé, yeah, it's clear if we want to Uto-Aztecan, at least in the way that I'm using it, that's Paquimé. There's not, it's that green component on the admixture graph. That's clearly there at Paquimé. I have another question about La Playa lies just outside of Trincheras and push back the dates there with long habitation and probably was the oldest dates anywhere. Yeah, we have individuals from La Playa. We have a pretty good time series from Trincheras and La Playa. Really old individuals and more recent individuals. And genetically, they're pretty much homogeneous, meaning we don't see change through time there. But like everything I've been talking about, it's going to get detailed, work as we move towards publishing the paper.
All right, brilliant. Well, if there's nothing else coming in, I suppose we can end it here. Thanks for a brilliant presentation, Jacob, and also for everyone attending and contributing to this talk. Just a reminder that this one's been recorded and it's going to go up on our YouTube channel probably by next week. And if you have questions asked, you could always post something on YouTube and I could catch it and send it to Jacob myself, or maybe you could send Jacob an email or whatever.
Yeah. So, I'm easy to find if you Google me. My email is just jacob.sedig@gmail.com. So happy to answer any questions.
Yes, excellent. And finally, just a reminder that we'll of course be doing a June lecture. It'll be by another coworker of Jacob and I's, Angela Huster. She's going to be talking about the Aztec conquest of the Toluca Valley. So it should be an interesting one. But otherwise, thank you, Jacob, and thanks everyone for showing up, and have a pleasant evening.
Thanks, everyone.